Hello everyone, my name is AJ Keefe, and in this presentation I'll be covering the paper PARL mediates Smac proteolytic maturation in mitochondria to promote apoptosis, published in the Journal of Nature Cell Biology. I want to extend a special thanks to the authors for their contribution to this field of research. And for this video, I recommend that my viewers pause the video at each new slide and read through the material and analyze the graphics before actually playing my commentary. And if you have any questions, please feel free to comment them below and I'll be sure to respond. And I hope you enjoy the video. Before diving into this paper, we need to briefly cover the process of apoptosis, as well as the specific activity of these three proteins, PARL, Smac, and XIAP. So this figure to the right shows us the general apoptotic pathway. The extrinsic pathway typically begins with the activation of the death receptor, the DR4 or 5, by its ligand, trail, which is TNF-related something ligand. So the activation of this death receptor recruits a death-inducing complex called DISC, for death-inducing signaling complex. The DISC complex contains a CARD domain, or a caspase recruitment domain, that recruits caspase 8 and promotes its proximity-induced cleavage and activation of caspase 8. Caspase 8 then bridges the extrinsic pathway with the intrinsic apoptotic program by cleaving the BH3 protein called BID. When BID gets cleaved, it dissociates from cytoskeletal components and translocates to the mitochondria right here, where it promotes the oligomerization of BAX and BAC, which forms a pore in the outer mitochondrial membrane called the MOMP, M-O-M-P, the mitochondrial outer membrane pore. This pore releases a toxic cocktail of proteins into the cytosol, and two very critical proteins that are released by the MOMP are cytochrome C right here and Smac. So cytochrome C induces the formation of the apoptosome, which is the caspase activation platform for caspase 9. Caspase 9 then goes on to cleave caspase 3 and 7. And a common mechanism of assaying caspase activation, as you can see in this graphic, is the amount of cleaved PARP or PARP. Don't confuse PARP with PARL. They have similar abbreviations, but they're completely different. So PARP cleavage by caspase 3 right here is a common proxy of caspase 3 activity, and we'll see it used in this paper. So cytochrome C, apoptosome, caspase activation pathway is actually constitutively inhibited by this other cytosolic enzyme called XIAP right here. So X-linked inhibitor of apoptosis. And it constitutively inhibits caspases, like caspase 9 and caspase 3. And it does it very well. So XIAP actually binds caspases and inhibits them. So any caspases that get activated by cytochrome C and the apoptosome are just constantly inhibited. Unless, and this is critical, unless Smac is also released into the cytosol. So Smac promotes apoptosis by inhibiting XIAP. And so Smac inhibits the caspase inhibitor. And so by the rules of double negatives, we can say that Smac is activating caspases indirectly, because we see Smac inhibiting the inhibitor of caspase 9 and caspase 3. So there's one more key player in this this whole ordeal, and this is and it currently doesn't fit into the apoptotic pathway in any recognizable way until this paper, and that's PARL, P-A-R-R-L. PARL is a mitochondrial protease that is known to be involved in remodeling mitochondrial cristae, and its mutations are associated with Parkinson's disease. And its mutations in mice causes a multi-system atrophy. So basically, the entire body just begins to degenerate, and it also so this PARL is critical for an organism's survival, and it's also critical for mitochondrial function because when it's knocked out, the oxidative capacity of the mitochondria just plummets. And we'll find out in this paper that PARL is also vital for the induction of apoptosis. And so our three main players are XIAP, which inhibits caspases, Smac, which inhibits XIAP to promote caspase activation, and PARL, which is going to be found in the mitochondrial membrane. And we'll discuss what PARL has to do with Smac in a second. So PARL, this mitochondrial protease that is known to be critical for mitochondrial function and also oxidative capacity and an organism's general survival, is actually poorly understood beyond its role in cristae remodeling. So the current paper was initially designed around simply identifying PARL substrates through an unbiased high-throughput screening assay. And they wanted to see what proteolytic PARL substrates might modulate apoptotic sensitivity. So in a general sense, they, at least initially, just wanted to better characterize PARL's cleavage activity. However, after screening for PARL substrates, they identified a very curious substrate that is highly important for the induction of apoptosis, and that was Smac. So their first discovery that provided the impetus for this research was that PARL cleaves Smac, a critical regulator of apoptosis. And they wondered if Smac cleavage by PARL was important for the process of apoptosis. So they looked closer at this cleavage reaction, and they noticed that cleavage deficient PARL enzymes or complete PARL knockouts caused Smac proteolysis cleavage to drop by about 80%. And they also observed that Smac cleavage occurred at its transmembrane domain. And without cleavage, Smac remained attached to the membrane of the mitochondria. And this was a big deal because that meant that without PARL, Smac was unable to diffuse out of the mitochondria when the MOMP, the mitochondrial outer membrane pore, was formed. And they observed that PARL deletion increased cell viability following apoptosis stimulation by about 30 to 50%. And it also reduced the amount of PARP cleavage, P-A-R-P cleavage, by about 90%. Remember, PARP cleavage indicates caspase 3 enzymatic activity, and the loss of PARL caused PARP cleavage to drop by 90%. So they further interrogated what's happening, and they found out that the reduced apoptotic sensitivity observed in the PARL knockouts was due to Smac remaining in the mitochondria. And thus PARL deletion prevented Smac release from the mitochondria. And furthermore, XIAP inhibition, which is what Smac normally does when it's released into the cytoplasm, was able to restore apoptotic sensitivity. So by experimentally inhibiting XIAP with siRNA or by Smac mimetics, which mimic Smac's XIAP binding, they were able to restore the cleavage of PARP and apoptosis induction. So this paper ultimately shows that PARL-mediated cleavage of Smac is critical for the entrance into apoptosis, and thus could have big implications for diseases such as cancer. This paper was originally based on this high-throughput, unbiased screen of PARL substrate specificity. And they conducted two cleavage product assays. First, they immunoprecipitated PARL, and they identified all of the proteins that co-immunoprecipitated with an enzyme-deficient PARL through mass spectrometry. And they found 45 proteins enriched in the PARL mutant cells, suggesting these proteins were binding PARL, but since the catalytic activity of PARL was inactivated by mutation, they simply remained associated with PARL. And they identified 45 proteins through immunoprecipitation, followed by mass spectrometry. And they also did a technique called CHA-Fredic right here. I'm not sure exactly how to pronounce it, but it stands for charge-based fractional diagonal chromatography. And this technique is able to separate N-terminal protein fragments by chromatography, and they found 46 N-terminal peptides that were significantly enriched in the wild-type PARL, but absent in the PARL mutant cells. So these 46 N-terminal fragments that were enriched indicate fragments that are normally cleaved by PARL, and when PARL was missing or mutated, they didn't show up in the CHA-Fredic analysis because they were still bound to their parent protein. And so the 5 proteins that were implicated by both techniques were most notably Smac right here. And we also see star D7, and TTC19. There's a couple more, but PGAM5. So these are proteins that were identified by both high throughput cleavage analysis. And also note PINK1. It's a known substrate of PARL, PINK1 is, although it was only observed in this co-IP analysis and not in CHA-Fredic, likely because its cleavage is dependent on membrane depolarization, but that's another story. But anyways, the identification of Smac by both approaches was probably pretty exciting because it's such a critical player in apoptosis. And so in light of that, they decided to look at Smac a little more closely and see exactly how PARL is mediating Smac activation. In order to understand the importance of PARL's cleavage of Smac, they utilized a PARL knockout cell line that instead had a flagged PARL enzyme knocked in. So this might sound a little paradoxical, but what they wanted to get rid of the endogenous PARL enzyme. Up here, you can see it's knocked out. So they could instead have a PARL enzyme that is tagged with a fusion tag like flag. And this allows for easy purification or immunostaining and for some other purposes as well. So in this western blot to the right, we have a western blot of Smac and star D7. So we're in these mock controls, we also have wild type controls, and we have the PARL mutated controls. So in this first lane, we have a wild type cell with wild type PARL. So this is a completely normal cell. We see a single very dark band at the size of Smac that corresponds to the size of a mature Smac fragment. So we see a Smac that is M, meaning mature. And in the second lane, we have a PARL knockout in a wild type cell. So it's a cell that's missing its PARL. And we see that the loss of PARL significantly reduces the amount of mature Smac, indicated by this little band right here. And instead, we're seeing 3 total bands. We see a faint band at the size of size of P, or pre-processed Smac right here. We see a little bit of it. And we see some I, or intermediate Smac right here. We see about 20% the amount of mature processed Smac when PARL is missing. Then we see the same pattern in these 2 lanes. So this is wild type, so it's PARL deleted, but it contains the PARL flag. So in this wild type condition, we have PARL, and again, we're seeing the same pattern. We see the flagged PARL enzyme is cleaving Smac into the mature fragments that we would expect. In the mutant flagged PARL, which lacks the enzymatic activity because it has this S277A mutation. This is a mutant PARL that's been flagged. And we see this also causes a phenotype that resembles the PARL knockout, which we would expect because it doesn't contain cleavage ability. So we see downregulated Smac processing, about 80% less mature Smac. When this is actually measured, we see about 80% less. And we also see downregulated star D7 right here and CLPB right here. So they're seeing that this PARL deficient cleavage enzyme is reducing the amount of processing of Smac, star D7, and CLBP. The next step was to identify if this cleavage was needed for membrane dissociation, because the consensus sequence for PARL, which was identified in the last slide, this sequence is found on Smac right along the transmembrane domain, suggesting PARL cleavage of Smac might be required for the dissociation of Smac from the membrane. And so to test this, they conducted another Western blot of soluble, so S means soluble, and P means membrane-bound Smac. And it's only until the pH is cranked up, which causes protein dissociation from the membrane, before Smac is solubilized. So we see the amount of soluble Smac in the PARL knockouts is almost non-existent. So there's very, very little mature Smac that is soluble, and most of the mature Smac that is around is still attached to the membrane. So we see in the membrane fractions from the Western blot in the P lane, we still see mature Smac still attached to the membrane. So even though it's still getting cleaved by some other enzyme, it's not getting dissociated from the membrane. So PARL is, we see over here that when PARL is wild type, there's plenty of soluble Smac that is at the mature size. So we have large full-sized Smac in the wild type condition. So PARL is not only needed for the cleavage of Smac into the mature form, but it's also needed for the dissociation of Smac from the membrane so it can be solubilized and subsequently released through the mitochondrial outer membrane pores. The next question at this point is, does PARL-mediated Smac cleavage and dissociation from the membrane actually have any functional consequences for Smac's role in apoptosis? So in these two sets of experiments, they are looking at PARP cleavage. So C-PARP is cleaved PARP, and that's, again, a measure of caspase activity. And cell viability was assayed by ATP concentrations and nuclear morphology. So in this top right graph, they observed that PARL negative cells, or PARL negative cells expressing an enzyme deficient PARL in green. So in green is enzyme deficient, or a PARL that can no longer cleave its substrate, and in red is just no PARL at all. And we see in these two PARL deficient conditions that there's significantly less cleaved PARP. About half as much PARP is cleaved compared to PARL wild type conditions in either the blue, which is complete wild type, or the yellow, which is PARL, is flagged PARL that's been knocked in. And this is, the condition is TNF-CHX. So this is remember, TNF activates the TNF receptor, which signals apoptosis through the DISC, the death-inducing complex. caspase 8 and then BID. And the addition of CHX is it inhibits translation and further promotes apoptosis. So basically what we're seeing here is that when PARP is deficient, because it's either completely knocked out in red or lacks its enzymatic activity, that the amount of cleaved PARP, caspase activity, is significantly downregulated. And the next thing the researchers assessed was a more broad symptom of apoptosis, and that's just cell viability in general. And they assayed this with ATP concentrations in this graph and nuclear conformation in the second graph following STS treatment, which is an inhibitor of kinase activity, basically inhibits most kinases and induces apoptosis, and actinomycin D over here on this figure to the right, which blocks transcription and also induces apoptosis. So again, we see the same pattern here in the PARL knockouts, which is going to be red this time. When PARL is knocked out, we see a increase in cell viability as measured by ATP concentrations. And we see decreased cell viability when PARL is present, either flagged PARL in yellow or wild type PARL in blue. And again, we see the same pattern over here. When cells are challenged with actinomycin D, the PARL that is flagged or the wild type PARL, if it's present, then the cells lose cell viability. When PARL is knocked out in red, we see increased cell viability. And so both results are suggesting that PARL-mediated cleavage of something is promoting apoptosis. And it's very likely that PARL's cleavage of Smac is the reason cells are not responding appropriately to apoptosis. But we can't quite make that connection yet because it's not been directly confirmed. And so they need to dig a little deeper. Since Smac detachment from the membrane required PARL, it's an intuitive next step to assess Smac's ability to be released into the cytoplasm following apoptosis induction and mitochondrial membrane permeabilization. And indeed, we see in this graph to the right that Smac localization in the cytosol, so this is the cytosolic fraction of Smac, which is what they're looking at, requires PARL and apoptosis induction. So in the blue and red, we see wild type conditions. In the blue, we see wild type conditions when apoptosis is negative. So no apoptotic induction right here. We see very, very little Smac localization in the cytosol. We see in the apoptosis inducing condition in the red bar, we see wild type cells induced into apoptosis with, I think it was STS. We see Smac is highly localized in the cytosol. Then we see the PARL knockout conditions. When PARL is missing in the yellow condition, which you can't even see the bar, when PARL is missing and there's no apoptosis, there's no Smac in the cytosol. That's not too surprising. What is surprising about this figure is the green bar. In the green, we see induction of apoptosis in PARL mutant cells, and there's almost no Smac in the cytosol. So Smac is not getting released from the mitochondria when PARL is missing. In the bottom figure, we see essentially the same thing, but in a Western blot format. So let's start with the whole cell lysate, so whole right here. We see the amount of mature Smac is indicated by this incredibly faint band right here. You can't even hardly see it. This is the amount of mature Smac in PARL KOs, so knockouts. There's very little Smac present. And in the mitochondria of wild type, in wild type PARL, we see lots of processed mature form of Smac in the wild type. And then we see a decrease when apoptosis is induced. And that makes sense because in this condition where apoptosis is induced in the wild type condition, we know that Smac is leaving the mitochondria into the cytosol. We also see correspondingly an increase in the cytosolic amount of Smac right here. So we see Smac is accumulating in the cytosol when apoptosis is induced in the wild type condition. What's interesting is in the KOs, the knockouts. So again, we see that triple band phenotype because PARL is missing in these KOs. So we see pre-processed right here, intermediate right here, and mature form Smac right here. And there's very faint amount of mature Smac. But what's probably most interesting is following apoptosis, the amount of Smac doesn't really change. We would expect Smac to be released during apoptosis induction right here. But we see it's essentially the same amount. In the cytosol, we see why, and that's because there's almost no Smac in the cytosol when apoptosis is induced. So when these knockout cells, apoptosis induction is not causing the release of Smac. So these results suggest collectively that PARL cleavage of Smac is needed for Smac's translocation to the cytosol following apoptotic induction. So just to hammer home this idea that Smac is not getting into the cytoplasm when PARL is mutated, they showed that Smac's primary interacting partner, which is XIAP, is not interacting with Smac when PARL is knocked out. So they're knocking out PARL, then they're precipitating Smac. So let's just focus on this one right here. So they're knocking out PARL, and then they're precipitating Smac, and you can see, and then they're staining for XIAP in the Western blot. So you can see when PARL is knocked out, the interaction between Smac and XIAP is vastly downregulated. So they don't hardly interact at all, and that's likely because there's dramatically less cytoplasmic Smac in its mature form. And interestingly, they found that HTRA2 interaction with XIAP is upregulated when PARL is knocked out. And so this is probably a compensatory interaction since Smac is no longer present in the cytoplasm. HTRA2 is probably taking on some of that burden and binding XIAP and cleaving it, because that's what HTRA2 is. It's a protease that's released. But also look down here, we see the same pattern. The mature form is vastly downregulated. But the main idea of this slide is that interaction between, we saw in the last slide that Smac is not getting released into the cytosol, and that has consequences, including this right here. We have less inhibition of XIAP by Smac. So the theory at this point is that the reason PARL knockout cells are less sensitive to apoptosis is directly because Smac is unable to get processed and released into the cytosol, where it inhibits and binds XIAP. So to prove this, they show that inhibiting XIAP by either siRNA down here or by a small molecule that mimics Smac up here, which is the BV6, both of these things restore apoptotic sensitivity. So in other words, they are artificially and experimentally doing Smac's job. That is, they are inhibiting XIAP. And that is what Smac is supposed to be doing, but can't do because PARL is mutated in these conditions, in the red condition. So in this top graph, we see the effects of apoptosis induction by STS, and what happens when cells are also treated with a small peptide BV6 that mimics the binding of Smac to XIAP. So in this first portion right here, STS alone, we see the usual pattern. That is, when PARL is missing, the amount of cleaved PARP is vastly downregulated. So the amount of caspase activity when PARL is missing is downregulated because Smac is not getting processed and released. We see, however, that when BV6 is co-administered with STS, we see the almost complete restoration of cleaved PARP. So the only difference between these two conditions is the presence of BV6. Smac is still sequestered in the mitochondria in this red bar, this red condition. The only difference is that they've done Smac's job and inhibited XIAP using this BV6 small molecule, and it restores the sensitivity. And we also see a similar pattern down here with the siRNA so we have XIAP siRNA. When it's absent, we see very little cleaved PARP, over here. In the PARL knockouts, we see almost no cleaved PARP, right here. However, when you add siRNA towards XIAP, we see tons of cleaved PARP, suggesting that the activity of caspase-3 is being restored by the inhibition of XIAP. So, and we can see this happens despite Smac still being in its mostly intermediate form. We see the restoration of cleaved PARP despite Smac still being unprocessed, largely unprocessed. So collectively, we can say that the loss of Smac processing by PARL prevents cytoplasmic localization and inhibition of XIAP, and restoring XIAP inhibition restores apoptotic sensitivity. Okay, so just a quick summary of what we learned. We learned that PARL-mediated Smac cleavage is required for cytosolic relocalization of Smac during apoptosis, and it's also required for the inhibition of XIAP. And disruption of this process significantly reduces apoptotic sensitivity. So when PARL is no longer able to cleave Smac, cells are no longer responding to apoptosis. However, this is restored when XIAP inhibition is restored. So altogether, I feel like this paper is pretty adequately tied up. It's tied up all the loose ends, and I'm left pretty satisfied with the general process. So I guess my only two questions are one, what proteases account for the residual 20% of mature processed Smac that remains after PARL deletion? And why are these Smac proteins still not capable of cytosolic relocalization? And my other question I'm left with is, does PARL promote or inhibit cell death over the long term? Because we know PARL is critical for an organism's survival, and on a molecular level, it's critical for the remodeling of mitochondrial cristae. But at the same time, it's critical for inducing apoptosis by releasing Smac. So in general, does PARL promote or inhibit cell death? And perhaps it's a time-dependent relationship, and I would guess that over the long term, it probably promotes cell death by the loss of mitochondrial integrity. But in the short term, it probably prevents apoptosis by withholding Smac in the mitochondria. But anyways, I hope everyone enjoyed the presentation, and please comment any questions you have, and I'll be sure to respond. And thanks for watching.